Just give me a pen.
Blue pen.
Okay.
Tell me your name.
Khushi.
Hmm?
Khushi Agar.
Khushi.
KH.
Gaukur spelling. G.
A O N K R.
Eraser से. Yusuf were present last class. Yes, right. He came late. I'll give this pen later. I thought you're shifting and giving us some chocolates or something. I know. You want AC? Put it on. Okay, close your textbooks, open your notebooks. Yuziko, where were you last class? Teacher, smile. Teacher with a smile. Why? Where were you? Udta Ifiru. Went somewhere far after exams. Then was at home. So why you didn't come here? Just simply. Okay, I'll open notebook. Give me a notebook for some time. Give. That's what. That's why you're writing on the last page. Even last class you did the same thing. Why? Where is your bio notebook? Is there any bio notebook in existence? At home. Pujek Dawalla. Then? Okay. But how you'll track down how much you're scoring in each and every test then? Yeah, I put at least once in a week you have surprise test, no? So, Lisa knows. She's smiling also. She's very happy that we have test. Yeah, I know. I can read your face. Okay. So here are some simple questions which you have to answer. Number one. Question number one. Yeah, of course. Okay. Question number one. Explain structure of nucleus. Explain structure of nucleus in prokaryotes. In prokaryotes. 1 mark. Second, write down a short note on cytoplasm. Write a short note on cytoplasm. 1 mark. Third question, name the largest isolated single cell. Name the largest isolated single cell. Next question. This question is for 1 mark. Next question. Based on shape and size, based on, not size, based on shape, name the different type of, name the different type of bacterias. Different type of bacterias. 1 mark. Next question, which is the last question. Write any two distinct feature of prokaryotic cell. Distinct feature of prokaryotic cell. Okay, so your time starts now. Just write whatever you know. Vidal más pues puede llegar. Daniela, the remote thing. Sister. They have a jacket with Galan Basla. And you're saying you're feeling hot. Take some inspiration from Lisa. If you are done or if you don't know the answers, you can give for correction. Done? Chapter Shakela. Is completed. काय नाही, no problem. क्वेश्चन्स के है आणि टेक्स्ट बुक असो. No, I'm not correcting it if I'm not coming here. आणि थोडे बच्चे बारीक झाले. आता करेक्शन. तो बरेला पेन्सिल आहे. Should I correct with my pen? And what will be the proof that you have given this test when you erase it? Then I'll correcting it with the pen. They have. Yes, anyone else? Yes, anyone else? Other questions you have not written? नेक्स्ट टाइम क्वेश्चंस मिस आउट झालेले तर कोणाकडून घे ओके yes what is the first question yes so what does nucleus in prokaryote is having a different or unique feature hmm yes it is not well defined number one why it's not well defined because it is not having nuclear envelope न्यूक्लियसच्या एक बाहेर एक envelope असता एक covering असता that covering is absent in this nucleus of prokaryotes that's why we are saying that it is not well defined okay what was the second question hmm? Yeah cytoplasm so what is the cytoplasm fluid matrix right fluid matrix of the cell okay main part or the main surface area of the cell and what is happening here chemical reactions majority of them takes place in the cytoplasm he दोन sentences बरोबर जाय आशिल्ले and also you can write down cell organelles are present on this cytoplasm okay next Yeah, largest isolated single cell is, yes, egg of an ostrich. Okay. Next, yeah, shape based on different bacteria based on the shape are what? Coccus, Bacillus, Spirulum, and a Vibrio. Okay. Last question. Yeah, only one person have written correctly. That is Skyla. So what are the distinct features of prokaryotes? Mesosome, plasmid, 70S ribosome. Okay, not well defined nucleus. These are the distinct म्हणजे किती आहे? Which is not common with the eukaryote, which is different. Okay, distinct म्हणता आपण आम्ही ज्यांना distinct word येता, that means you're supposed to write down only the features which are relevant to prokaryote, not any other cell, that is eukaryote. Okay, similar असतं ते बरोबर ना. Is this clear to everybody? Yes, you might write that prokaryotes have cytoplasm. That is correct. Prokaryotes and cytoplasm असतं, but is it the distinct feature of prokaryote? No, because cytoplasm is there in the eukaryotes as well, right? Yes, is this clear to everybody? Okay, thank you. Last time, yes. So आतापर्यंत आम्ही किती पहिला we have seen. Okay, the pattern of my teaching is I give some keywords म्हणजे full chapter on खंयचे खंयचे words याद दवरपाक जाय that I'm giving on the very first page आणि तेजे वयल्यान कितें करपाचें आमी if any one of the word you have not understood that means तुमकां तो concept समजुना so you're supposed to ask me about that particular keyword आणि हे जर जेन्ना questions येता पळय आणि हे questions answers बरयतना if you have not included these words that means you will not get full marks. Okay, so here words aspakut these are very important terms from this particular chapter. Okay, is this clear to everybody? Yes, Vidal, I can't see you. Yes, what is plasmid, which is the key character of the prokaryote. Circular DNA म्हणजे usually how DNA is present or no. Yes, like a rod. That is the normal DNA, right? So you draw DNA like this, right? This is the proper way of like normal way of drawing any DNA. Circular DNA कसं असतं? Okay, join असतं. वो joint ना. Okay, that is the difference. This is linear DNA. याला किती म्हणता? Linear DNA. This is circular DNA. Okay, this is clear to everybody? Okay. Now, unicellular cells असता आणि also there are multicellular or not cells, organisms. Unicellular organisms, multicellular organisms. Also, we have learned here about two scientists' name. कोणतं? Antonio Leeuwenhoek आणि दुसरा असा तो Robert Brown. Leeuwenhoekने किती केलंय? So Leeuwenhoek described live cell for the first time, and Robert Brown he described or he discovered about the nucleus. आणि कसा Robert Hooke जो आम्ही 9th शिकलेली, and he was the first person to actually study about the cells. पण त्यांनी किती केलंय? Dead cell study केलंय. Okay, is this clear? Now, so there is one concept called as cell theory, आणि ह्या theoryच्या according किती किती शिकले आम्ही that we organisms are composed of cell or the product of cell, and we know everything is coming from the one pre-existing cell. एक पहिली cell आशिलो तो divide जाऊन modification जाऊन किती झाला, we have got so many varieties of species. Okay, मागे overview of the cell म्हणजे सेलान किती किती present असा. Number one, there is cell wall, and cell wall is present only in the case of plants and few bacteria. Okay, मागे what we have is cell membrane or plasma membrane. This is present in both plants and animals. Then what we have, nucleus. Nucleus well defined in the case of eukaryotes, not well defined in the case of prokaryotes. Then we have cytoplasm, which is the main compartment ज्या जागेवर सगळे organelles असतात आणि ज्या जागेवर सगळे chemical reactions जातात. Then we have membrane bound distinct structure and non-membrane bound structures. So membrane bound, non-membrane bound हेंका किती असतले? There will be covering, there will be an envelope for all these organelles, आणि हे जो नसता तेंका किती असा? There is no covering or no envelope which is present. Okay, is this clear? So कोणाक envelope ना or non-membrane bound कोण असा? फक्त दोन, रायबोसोम आणि centrosomes. Henceforth, I will not ask you any questions. Okay, I'll not waste time also because I feel response mehna, it's fine. I'll just finish with everything and I'll go. इतले 2-3 क्वेश्चन विचारले उद्याच्यान, ते बी इतले सिंपल क्वेश्चन्स. हांगा आन्सर्स दिसता स्टील तुमका तोंड उघडपाक सुद्धा जायना. So it's okay, I won't waste my time henceforth, and I'll not waste my time in talking stupid things also. I'll just finish off everything and go. Okay, now next sizes आम्ही compare केला between unicellular आणि multicellular. We have 3 examples here, 2 examples here. 3 example कोण कोण? Mycoplasma, bacteria आणि ostrich, which is the biggest single cell. Then we have multicellular example किती असा? Human red blood cell, which is 7 micrometer, and we have nerve cell, which is the longest cell which is present. And shape, you know, shape can be different. It can be disc-like, polygonal, columnar, cuboid. It can be thread-like structure, or even irregular shapes of the cell are present. Okay, then what we have, function of the cell. Function of the cell will be changing depending on what function, where they are present, where they are located. Okay, then these are the diagrams which you have in your textbook. Then we started with the prokaryotic cell. Prokaryotic cell याच्या under organisms असा bacteria, blue-green algae, mycoplasma and PP yellow. One distinct character is that they are smaller in size and they are replicating very fast. This is revision from the last class. Okay, म्हणून इथले बेगीन सांगता हांव. Okay, you can watch the recorded video. Fine. Then, so we have 4 different types, or 4 different, you can say, bacteria depending on the different types of shapes. That is Bacillus, Coccus, Vibrio and Spirillum. Okay, next was about prokaryotic cell consists of what? Tumka wo question can be asked for 3 marks or 5 marks to write down about all the compartments or all the structures which are present in the prokaryotic cell. So prokaryotic cell and kiti kiti asa? It is having cell wall, cytoplasm, nucleus, plasmid, very distinct structure, which is a small circular DNA. होते काढला पय तसं small circular DNA असता. Ribosome, which is 70S ribosome. What is this S? S is the unit. Okay, जसे आम्ही 30 meters, 30 kilometers, kilometer, meter use करतात पय तसे हे किती unit. Okay, then mesosomes, these are the very special features of prokaryotes. So ज्यांना distinct features विचारले त्यांना आम्ही किती किती बघितली. We'll be writing about plasmid, 70S ribosome, and also about mesosome. You can also write down about the not well defined nucleus. Nucleus is lacking nuclear membrane. Yes, तुम्ही बरोबर शकता. Okay, next what we have is about cell envelope. ओके. सेल एनवलप और सेल कवरिंग विच इज प्रेझेंट हिअर, तिथून 3 डिफरंट लेयर्स असतात. व्हॉट आर द 3 डिफरंट लेयर्स? ग्लायकोकॅलिक्स, सेल वॉल, प्लाझ्मा मेम्ब्रेन. सेल वॉल पहिली ऐकला? होय. प्लाझ्मा मेम्ब्रेन ऐकला? होय. So there is one more layer called as glycocalyx, which is the outermost layer. So कशा असतले? It will be like glycocalyx, मागीर सेल वॉल आणि मागीर plasma membrane. Okay, but in the case of animals, what will be missing? Cell wall will be missing. Yes, is this clear to everybody? Any doubt? आणि यांच्या भितर किती असता? वेगळे वेगळे chemicals present असता, and this chemical composition will differ according to which species they are present in. Okay, म्हणजे आमच्या एनिमल्सा भितर वेगळे केमिकल्स आसा, प्लांट्साच्या भितर वेगळे केमिकल्स असतले. ओके, दिस इज डिपेंडिंग ऑन what type of species it is. Then all these things together, they are doing very important function, that is they are acting like a protective unit. They are protecting the cell from the external environment. Okay, then number 1 is glycocalyx. Glycocalyx is the outermost layer. Can you see here what dark green asato? That is the glycocalyx. Okay, so glycocalyx composition kiti asa? It is thick. Okay, it is thick, and again here you will find two types. This is A and B. Okay, it can be a slime layer or it can be a capsule. Slime layer आम्ही केन्ना म्हणता? जेन्ना it is very loose shape. Okay, the covering is not very hard, it's very loose. That time आम्ही किती म्हणता? Slime layer. आणि capsule केन्ना म्हणता? When it is very thick and it is very tough. Yes, this is about glycocalyx. Chemical composition तेजे vary करले again depending on what type of organism in which they are present. Yes, is this clear to everyone? किती doubt असा हांगा? ना. Second is cell wall. So cell wall याच्याबद्दल आमका थोडे किती तरी खबर असा, that is it is giving shape to the organism, right? Shape दितानी, cell wall is very strong. It's like almost like acting like a skeleton. Okay, skeleton ना जाल्यार body shape उरना पय तशें हे. Okay, then a strong structural support it is giving so that it can prevent the bacteria from bursting or collapsing. ओके, सो सेल वॉल जर नाशिल्लो न्ही आनी जर बॅक्टेरियाच्या और प्लांटाच्या मदीं जर खूब उदक लायक एक्वियस कंडिशन भायर जर चड आसा जाल्यार कितें जाता? उदक सेलाच्या भितर भरपाक लागता, रायट? आमी एका सायडीन एनिमल सेल घातले, एका सायडीन आमी प्लांट सेल घातले. हू विल बर्स्ट फर्स्ट? दी एनिमल सेल, नॉट दी प्लांट सेल, बिकॉज प्लांट सेलान कोण आसा? Cell wall आसा. So this is actually protecting it, okay, from bursting and from collapsing. Fine. Plasma membrane, cell membrane. This is selectively permeable. This word is very, 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 very important. जेन्ना जेन्ना तुमका cell membrane or plasma membrane बरोवपाक येतलो, for example, write a brief note on plasma membrane, cell membrane. The very first thing you are going to write that it is selectively permeable. Selectively permeable म्हणजे कितें? ज्यो गोष्टी सेलाच्या भितर required असता, only those things it is going to allow to come inside, and the things which are not required inside, that it is allowing to go outside the cytoplasm. Okay, so भितर यो भायर वचप, everything is guarded by this plasma membrane, and hence we are calling it as selectively permeable. Okay, is this clear to everyone? किती doubt असा हांगा? And this is actually the layer जो किती करता? It is interacting with the outside world. Outside world म्हणजे whatever content that is supposed to come in or go out, ते interaction कोण करता? Cell membrane or plasma membrane. This membrane is similar in structure in that of the eukaryotes. So eukaryotes and plasma membrane is some more or less similar as compared to the prokaryotes. Okay, is this clear? Fine. So please highlight this: glycocalyx, cell wall, plasma membrane. In plasma membrane, highlight selectively permeable. Here we highlight slime layer and a capsule. Okay. Now, next is about staining. स्टेन कसलो यूज करता हांगा ग्रॅम स्टेन. सो ग्रॅम स्टेन घेता आणि बॅक्टेरिया स्टेन करता. इफ द बॅक्टेरिया इज पिकिंग अप दिस पिंकिश कलर then it is gram negative bacteria, and if that bacteria is picking up this violet color then it is gram positive bacteria. इतलंच. So this is differentiation in bacteria based on staining. ओके. जर पिंक कलर घेतला तेन्ना कितें म्हणटा आमी ग्राम नेगेटिव्ह बॅक्टेरिया, आणि जर वायलेट आसा जाल्यार इट इज ग्राम पॉझिटिव्ह बॅक्टेरिया. ओके. येस, यू कॅन हायलाइट दिस ग्राम पॉझिटिव्ह and gram negative in your textbook. नेक्स्ट इज अबाउट मेसोसोम्स, आणि आम्ही लास्ट टाइम कितें डिस्कस केलं की मेसोसोम इज वन ऑफ द डिस्टिंक्ट कॅरेक्टर ऑफ प्रोकॅरियोट्स. ओके. कॅन यू सी मेसोसोम्स एनीवेअर हियर? येस. पळय. सो दीज आर कॉल्ड एज इनफोल्डिंग्स. आणि ऑलवेज रिमेंबर, जेन्ना जेन्ना खंयची इनफोल्डिंग सेलान जर प्रेझेंट आसा जाल्यार ती इनफोल्डिंग्स कित्याक हेल्प करता? टू इनक्रीज द सरफेस एरिया. ओके. आणि खबर आसा, मोर द सरफेस एरिया इज ऑलवेज बेटर, राइट? सो हे मेसोसोम्स कितें करता? दे आर इनक्रीजिंग द surface area. Okay, so we'll learn more about it. So they are what? Formed by extension of plasma membrane. Okay, plasma membrane extension. Okay, so they can be of various types. Number one is vesicles. Vesicles म्हणजे goal, like a ball. Okay, round like a ball. In biology, always remember, jenna jenna मी vesicles म्हणटली पळय, that means we are talking about a structure which is circular in shape. Okay, vesicle म्हणजे कितें? Circular in shape. Tubular म्हणजे tube like shape, or it can also be a lamellae. Okay, fine. So it can be vesicle, tubules, lamellae. Lamellae जो word असा पळय, वो चड in detail we will learn in physiology part. Okay, त्यांना वो चड बऱ्या भशेन समजतलो. Fine. Now, what are the role of these mesosomes? 1, 2, 3, 4, 5 roles असा. So this question can come for 2 marks. Okay, this question can come for 2 marks, and this diagram is actually not there in your textbook. This is only for you to understand ki हे mesosomes खय असता. Okay, so number 1 function कितें? They help in cell wall formation. Number 2, DNA replication and distribution to daughter cells. Daughter cells म्हणजे कितें? हो एक cell असा. आता हो cell divide जावपाचो आसा. So now it is forming 2 daughter cells, right? वो parent or mother cell, these are daughter cell. Okay, so या daughter cells DNA जो असता पळय, which is present in the nucleus, वो DNA equally पावपाक जाय, and that is ensured by whom? Ensured by mesosomes. Okay, next is they help in respiration. Next is secretion process. खूपशे chemical secretion जाता which are required for many processes, and that secretion is done by this mesosomes or controlled by this mesosomes. Magir, of course, जे आमी important role सांगलेलो, that is increase the surface area of the plasma membrane and enzymatic content. Is this clear? Mesosomes. So please highlight these things: vesicles, tubules, lamellae. आणि हे functions जे आसा पळय ते 1, 2, 3, 4, 5 number करा. Okay, just number that. एक sentence कशा असतले न्ही, so just number it 1, 2, 3, 4, 5. उदक जाय. Done? Then next about chromatophores. So chromatophores म्हणजे कितें? These are membranous extension into cytoplasm. Just extensions which are present in the cytoplasm, present in some eukaryotes like cyanobacteria, and it is containing some pigment. So here can you see green color thing, that is nothing but chromatophores, and this is one of the character of the prokaryotes. Okay, इतलेच ह्याचे फक्त 3 sentences दिला. Next is about surface structures in prokaryotic cell. Okay, first highlight this flagella, pili, fimbriae. Flagella, pili, fimbriae. सगळ्यात पहिली म्हणजे 3 स्ट्रक्चर्स तुमका आयडेंटिफाय करपाक कळपाक. द लॉंगेस्ट वन इज फ्लॅजेला, त्याच्यान मातसो लहान इज फिम्ब्रिए, आणि सगळ्यात लहान विच इज लायक स्मॉल हेअर लायक पार्टिकल तेका आमी कितें म्हणटा? पिलाय. फ्लॅजेला सगळ्यांनी ऐकला, राईट? So this is the difference between these 3 surface extension. Surface extensions म्हणजे कितें? It is always present on the surface of the organism. भितर present असपाना ते on the surface of the organisms. Okay, so सगळ्यात पहिले कितें पळयता आमी? Flagella. Okay, so this flagella bacteria can be motile. Motile म्हणजे can move from one place to another. Non-motile म्हणजे they are stationary. Okay, and this is actually basically the main function of this flagella. Flagella जर present असा, then that bacteria or that organism is motile in nature. म्हणजे ते move करपाक शकता. Okay, so motile, they have thin filamentous extension from their cell wall called as flagella. Okay, मागीर नंबर आता हे फ्लॅजेला 1 असा, 2 असा, 4 असा, कितले असा, that is completely dependent on what type of species it is. Okay, for example आता हांगा कितले असा, 1 2 3 असा, it can be 1, it can be 2. आता हे एकाच जाग्यार असा, but it can be here, it can be here, it can be here, it can be anywhere. Okay, depending on तांकां कसे कितल्या फास्ट वचपाक जाय आणि कशे मूव करपाक जाय. Okay, then 3 parts असता of this flagella. The longest one, tail-like structure, is the filament. So this is filament. Attachment करपाक help करता hook. आणि जे भितर मातशे embedded असा till plasma membrane or cell wall, till cell wall जे embedded असा तेका आमी कितें म्हणटा? Basal body. So what are the 3 parts or 3 sections of flagella? Filament, hook, basal body. Okay, please highlight these 3 words: filament, hook, basal body. The next about pili. So what are these pili? Again, they are surface structures which are present in some of the bacteria, but they are not playing any role in its motility. Okay, पिलाय आसा म्हणजे ते मूव करतले अशें काय तेजी गॅरंटी आसना. दे आर जस्ट प्रेझेंट. ओके, पिलाय आर इलॉंगेट ट्युब्युलर स्ट्रक्चर. ट्युब्युलर म्हणजे ट्यूब लाईक स्ट्रक्चर मेड अप ऑफ सम स्पेशल प्रोटीन्स. दॅट्स इट. हे फक्त 3 सेंटेन्सेस दिला अबाउट पिलाय. देन अबाउट फिमब्रिये. स्मॉल ब्रिसल लाईक फायबर्स. ब्रिसल लाईक म्हणजे कितें, बारीक ब्रशाक जशे बारीक हे असतात पळय तशे. ओके, very small, they are very small, and in some bacteria they are helping in what, आता flagella help करताले move करपाक, and fimbriae is completely opposite, they are actually helping in attachment. ओके, होस्टाच्या बॉडीचेर अटॅच करपा और खंयचो स्टोन आसा जाल्यार स्टोनाक अटॅच करपा. सो दिस इज completely opposite than the flagella. Flagella move करपा help करता. Fimbriae कित्याक help करता? It is helping for the attachment. Okay, so if they ask you 1 mark question different between flagella and fimbriae. Okay, एक तर size असतली, flagella are bigger, fimbriae are smaller, like you can say small bristle like structures. And second you can say what, flagella is helping in the movement of the organism, wherein this is helping in the attachment purpose. Okay, is this clear? किती doubt असा हांगा? Then next about ribosomes आणि inclusion bodies. So ribosomes, here we said it is 70S and S stands for Swedenborg's unit. Okay, S full form Swedenborg. This is not given in your textbook. Just extra have घातला because so that it's a unit of हा म्हणता पण त्याचा फुल फॉर्म सांगना म्हणून. Okay, it's Swedenborg's unit. आता they can be 15 nanometer to 20 nanometer in size, and the ribosome is divided into 2 structures. Wordlow part is called as the large subunit. Ductal part is called as the small subunit. So this is together is a prokaryotic 70S ribosome. आम्ही त्याला separate करू शकता. The bigger one or the bigger subunit is called as 50S. Smaller subunit is 30S. 50 plus 30 is how much? 50 plus 30. 50 plus 30. 50 plus 30. 80. But anga total आम्ही किती बरयता? 70S. So 10S खंय वयता? This is by the way biology maths. Okay, you can't say large subunit plus small subunit will give you the value of the whole ribosome. No, you cannot say that. Okay, because हांगा 10 जे आसा पळय ते खंय तरी लॉस जाता. That is a different topic. तो आमकां सद्या हांगा नाका. Okay, so large कितलो आसा? 50, small आसा 30, but together it is 70S. Okay, is this clear? Then, so these are very important role of ribosome. These are site of protein synthesis. जे proteins सामके important असता पळय ते synthesis कोण help करता आमकां? Ribosomes. आता if there is only one unit here, it is called as ribosome. More than one असता पळय तेका आमी कितें म्हणटा? Polysome or polyribosome. So this is called as polysome or polyribosome. This is the last part which is given here. Okay, is this clear? So this is one is ribosome, this is polysome or polyribosome. If they ask you what is polyribosome, what you are going to write? Presence of more than one ribosome is called as polyribosome or polysome. Okay, yes. Now last part, that is inclusion bodies. So inclusion bodies and किती असता? Reserve material or reserve food is present in this inclusion bodies. It is stored in the inclusion bodies and they are just scattered here and there in the cytoplasm. So हांगा पळोवंक शकता तुम्ही. This is inclusion body, inclusion body, inclusion body, inclusion body, inclusion body. They are just present randomly here and there. Okay, मागीर they are not bound by any membrane system and lie free in the cytoplasm. So तेका covering अशें कांयच ना. Okay, पहिले न्ही आमी membrane bound, non-membrane bound. So these are non-membrane bound. कोण non-membrane bound कोण? दोन structure. एक आशिल्लो ribosome, दुसरो is inclusion bodies here in case of prokaryotes. Okay, मागीर एक्झाम्पल ऑफ दीस इन्क्लुजन बॉडीज कोण आसा 4 आसा एक्चुअली हांगा 3 हांगा दिला दॅट इज फॉस्फेट ग्रॅन्युल्स सायनोफायसी ग्रॅन्युल्स एंड ग्लायकोजन ग्रॅन्युल्स आणि फोर्थ एक्झाम्पल इज gas vacuole. Okay, gas vacuole which is found in blue green and purple green photosynthetic bacteria. This is about prokaryotic cell. Prokaryotic cell 5 मार्का खातीर जर विचारले everything in detail without missing anything you have to write this. Is this clear? किती doubt असा? Just go through it once. I'm giving you 2 minutes. हे 3 चॅप्टरचे नोट्स आसा. Yes. Okay, now for next 10 minutes, we are discussing about eukaryotic cell and then we'll stop. Okay. So, eukaryotes, example कोण असा तिथून? Protists, plants, animals and fungi. These all are included in eukaryotes. What are the unique feature of eukaryotes? So the unique features म्हणजे हे features prokaryotes असपाक ना. Okay, so what are the unique? See, असे जे questions असा पळय, हे eukaryotes directly textbookan mention केला. But prokaryotes directly textbookan mention करू ना. That's why I am telling you that. Okay, so that तुम्ही तकलेन ते मार्क करून खंय तरी दवरपाक जाय की प्रोकॅरियोट्साचे युनिक फिचर्स कितें ते. ओके, कित्याक की सांग हेजे जर आसा न्ही जाल्यार there are some teachers who can ask you about the unique characters of the prokaryotes as well. Okay, so we have to be prepared for anything that is being asked. So हांगा कितें आसता extensive compartmentalization म्हणजे कितें prokaryote is like a 1 BHK room, 1 BHK flat आणि हो this is a huge bungalow. हेका खूप कितें आसा compartments आसा that is in the eukaryotes. Second important, there is presence of nuclear envelope on the nucleus. म्हणून आमी तेका कितें म्हणटा well defined nucleus म्हणटा. Okay, because it is having what? Nuclear envelope. Third thing is that there are complex, सामके difficult to understand, difficult to study दोन कितें म्हणटा ते processes असता. एक म्हणटा ते locomotory. Locomotory means movement. And then cytoskeleton. Cytoskeleton म्हणजे okay, we'll learn about cytoskeleton in detail. Okay, don't worry about it. But these two processes are very complex here in the eukaryotes. Okay, but last कितें आसा genetic material here is organized into chromosomes. Very important point to note down. हांगा कितें आसा chromosomes present आसा. Prokaryotes chromosome हो structure मेळना. Okay, this is actually called as packaging. For example, in humans, almost 2.2 meters long DNA असता. कितलो? 2.2 meters. इतलो लांब कितें असता? DNA असता. And in all the cells, and this 2.2 meters is packed in a very small nucleus in the form of chromosome. तो कसो असता पळय, that is a different chapter you have in your 12th standard. ताका packaging of DNA म्हणतात. Okay, is this clear to everybody? 4 points are clear. Compartmentalization, nuclear envelope, complex two processes, and the last one is presence of chromosome. Yes? आता eukaryote सगळे cell same असता अशें ना. Plant cell is different and animal cell is different. So जेन्ना आमी cell वो chapter 2 हितून divide करपाक सोदता पळय, we are going to divide it in this form. Cell, prokaryotes, eukaryotes. आणि eukaryotes कितें करता आमी मागीर plant cell आणि animal cell. Okay? Yes, this is the overview of this full chapter. हेच्या मागीर फुडें कितें 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 add जायत रावतलें. Fine? Now, so what we have? Major difference between plants and animals. So there is presence of cell wall, plastids, plasmid दुसरे, plastid दुसरे. Plasmid कितें आशिल्लें? Circular DNA आशिल्लें. This is plastids. In 12th standard, you will be having something called as microsporophyll, microspore, microsporangium, microsporangia, microsporophyte, microsporangia अशी करून करून 8 का 9 कितले तरी मायक्रो अशे टर्म्स आसा. So please remember की biology अशे टर्म्स आसतले जे similar sound करता but its meaning is different. If they ask you about the plasmid and if you write about plastid, then it is wrong. Okay, so plastid is present in plants. Plasmid is present in prokaryotes. So हे असो जेन्ना आमी हांव सांगता पळय तुमकां फ्लो चार्ट करपाक, ह्या फ्लो चार्टान कितें येतलें? ओन्ली द युनिक कॅरेक्टर्स. सो फॉर एक्झाम्पल हांगा कोण येतलो? मिझोझोम येतलो. ओके, नॉट वेल डिफायन्ड न्यूक्लियस. मिझोझोम येतलो. प्लाझमिड येतलो. 70S रायबोझोम येतलो. Okay, and hanga you can write down for eukaryotes. First you can write down the 4 points which we just discussed, and after that you can differentiate into plant and animal. And a plant and animal specific कितें म्हणटा ते features then you have to write. फक्त distinct common असा ते बरोवप ना, जे distinct असा different असा ते बरोवप. This is a homework for you. Okay, not now, but जेन्ना हो chapter सोंपता पळय, that time last thing you are going to draw this. Okay, notes आणि इल्लोसो space सोडटा. Okay, is this clear? Yes, any doubt? Fine. So plants आणि कितें आसा? Plastid आसा, cell wall आसा, large vacuole is present. Vacuole म्हणजे किती? Empty space. Okay, large vacuole is present, and all these things are absent in animals. Then centrioles are absent in plants, but centrioles are present in animals. आता हे centrioles म्हणजे किती? We'll learn in detail next. असा इथून या चॅप्टरचा. Centrioles are actually helping in division. Okay, fine. That we'll learn in detail. So this is plant cell. This is how we can differentiate. ज्यांना हांगा vacuum कसो असता पळय empty space, that is actually the vacuole. आणि this is almost you can say 80 to 90% of the cell इथूनच वता vacuolan वता. But हांगा तुमका vacuole दिसता? No, it's not there. Then again you'll have what? Cell wall. Can you see cell wall here anywhere? Yes, this one. Cell wall, right? Hangi ashtelo plasma membrane. Fine. Yes. Is there difficulty asa? I'm assuming the difficulty now and these things. Complex, locomotory आणि cytoskeleton. तो कळोना. Locomotory म्हणजे movement करपाक आमका पाय बी असता पळय. पण prokaryotes पाय बी असता ना. They are having some simple flagella or some structures like that. So that is complex. पाय कसो चलता, त्याचो खंयचो neurons खंय attach आसा, हे सगळे शिकप is a very complex process न्ही. So आणि cytoskeleton that will learn in detail. तो मी त्यांना सांगता कितें तो सारको exactly. Okay, that is there. पुढे असा एक बारीक बारीक पॅराग्राफ असा सगळ्यांचे. Okay, so आणि एक बारीक सो topic will just cover up here about the cell membrane, plasma membrane. Only the composition we'll learn today. बाकीचे मागीर पोवया. Okay, so composition, cell membrane, plasma membrane आणि किती किती असा? Number 1 is cell wall असा पयली नाही. किती असा? Lipids असा. ते बी कसले lipids? Phospholipids. Okay, then proteins असा. दोन टायपाचे प्रोटीन्स असा, इंटग्रल आणि पेरिफेरल. इन डिटेल कळटले. ओके, मागीर किती असा? कोलेस्ट्रॉल असा आणि carbohydrates. So all these compositions or chemicals are present in cell membrane or plasma membrane. आता सगळ्या प्लाझ्मा मेम्ब्रेन स्पीसीजान असतले असे ना, आणि सगळ्यांची कशे असतले proportion will be different. If this is in some animals you can say 20% lipids असतात, थोड्या हितून 50% असतले, थोड्या हितून 80% असतले. Okay, depending on what type of species it is, यांचे concentration आणि equation change जात राहतले. Okay, then main composition किती? दोन, लिपिड आणि प्रोटीन्स. मेजर लिपिड कसले असा? फॉस्फोलिपिड. फॉस्फोलिपिड म्हणजे किती? फॉस्फेट प्लस lipid, that is phospholipid. Okay, so they are arranged in bilayer. Very important term, bilayer. Phospholipid is important keyword here, and bilayer is also one important keyword. Okay, then so lipid ache arrangement कशे असा? So lipid is the layer. वो आता plasma membrane आम्ही करता पळय, तो जर आम्ही असं zoom केलो, if this part we zoom it, then we get a structure like this. Okay, वो जर आम्ही zoom करून पळयलो नी plasma membrane, cell wall असा भायर आणि वो असा glycocalyx. This is your plasma membrane or cell membrane. वो आता आम्ही किती करता? Zoom करून त्याचं structure पळयता. जेन्ना zoom करून पळयता पळय तेन्ना we see a bilayer. This is one layer, this is second layer. Two layers are present. Bilayer of what? Phospholipid bilayer. Okay, आणि वो कोण? This is one phospholipid. Okay, so this is वो circular जो असा पळय तेका आम्ही किती म्हणता? Polar head. Okay, circular is what? Polar head which is present outer sides. Okay, outside असा नी, आणि मधी किती असा? In between you have hydrophobic. Hydrophobic म्हणजे water hating, right? Hydrophobic tail which is present in the inner part. Okay, आता हे arrangement कित्याक असा? So polar head is water loving or hydrophilic आणि tail is hydrophobic. So तुमका खबर असा outside the cell or inside the cell दोन्हीकडे उदकाचे content सदात किती असता? चढ असता. Water content is more. So whatever that is moving from here, it has to undergo this hydrophobic layer. Okay, so कशा असं it is not easy थंयच्यान pass out जावपाक. म्हणून we are saying that this is a selectively permeable membrane. हेका कितें जातलें मदीं मदीं कोण आसतले बारीक बारीक channels आसतले बारीक बारीक tunnel आसतले लिटरली थंय एक पास आसतलो that only if you are required you will be allowed inside ना जाल्यार तुमकां allow करपाना. That's why this type of arrangement is very very important. Okay, so do one thing, just draw this bilayer काय ना असे गोल 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 काढून you can draw 4 or 5 like this and just label them. We can label this as polar head and label this as hydrophobic tail, polar head. This is a lipid bilayer. Also write down it's a lipid bilayer. Okay, so we'll stop here for today. Next class, we are going to discuss about more detail about this composition and structure of plasma membrane. Okay, please revise everything at home. Hardly 5-10 मिनिटं लागतील सगळं रिवाइज करपाक. Yeah, bye.